And our final speaker, by way of introduction, is Dr. Diana Castañeda-Cortés, the winner of the Postdoctoral Award. And she's obviously currently a postdoctoral fellow at the Ecotoxicogenomics and Endocrine Disruption Laboratory, led by Dr. Valérie Langlois at INRS in Quebec. She completed her PhD a few years ago, and I hope I pronounced this correctly, at the Biología de Desarrollo 2B in Tech laboratory in Argentina under the supervision of Dr. Juan Fernandino and Dr. Langlois. During her doctoral research, she studied the genetic and endocrine aspects of sex determination mechanisms and how they're impacted by climate change in aquatic species. Currently, Diana is working on the role of the enzyme that we heard already a bit about, steroid 5-alpha reductase type 2, in early development and reproduction in amphibians. And we look forward to her talk entitled Roles of Steroid 5-alpha reductase type 2 on gonadal development and reproduction in amphibians. Diana, the floor is all yours, and I'll just stop sharing here.
Okay.
Sorry, just give me 1 second.
Yes. So we are there. Yes. You see what my screen? Yeah. Are you seeing my screen?
Not yet.
No.
Sorry.
Yes, we see it.
You see it? I can, but
Okay.
that's okay then.
Yeah,
Okay.
yeah, well, I see it.
Okay, good.
Okay, thank you.
Good. Go ahead, Diana.
Well, let me, I don't know what, okay. Okay, that way, yes. So thanks, Ian, for the introduction and thanks for this opportunity to see up. So yes, like Ian say and Valerie say, I will present the role of the steroid 5-alpha reductase type 2 in the gonadal development and the reproduction of amphibians. This is not working. Yes, now it's working. So Valerie already did a nice introduction about this problematic of this subject in endocrine disruptions, but I will want to start to maybe take a little view about the different types of these enzymes and their roles in the individuals. So as already she said, the type 2 is mainly in the synthesis of the DHT, the 5-alpha DHT, but also the type 1 can have a role on this synthesis. As well, the type 1 have a role in the metabolism of the neurosteroids, the allopranolone, in special, that is more for the behavior. And then also the type 3 is mainly the metabolism of the glycosylation, the protein glycosylation, and the beta type, that is producing another type of androgen, that is the 5-beta dihydrotestosterone, that is mainly in the regulation of the inactive metabolites and the precursors of the bile acids. As you see, there is these enzymes are involved in many types of things in the body, but in this presentation, we are focusing the 5-alpha DHT and in the HRD type 2. So that we know in humans is that this hormone have a higher affinity for the androgen receptor than for testosterone. And we know from the studies of the knockout or the the lack of the HRD 5-alpha 2, that is generate a genetic disorder in humans that the normal that the individuals are genetical male, but with the presence of female or ambiguous external genitalia, as well infertility and many things related with this. As well, like I already Valerie explained a little, the overreactive or overexpression of this enzyme produce different things like the prostate cancer and the androgen and the alopecia. So all this the information that we have from human and why it's important this enzyme. But what happened in frogs, that is our interest here. So in frogs, this hormone is the most potent androgen. And we know this thanks to the studies using different endocrine disruptions, like I already Valerie introduced the finasteride. And we know that when we use the inhibitors of this enzyme, we can produce different phenotypes, like in Bufo, we have sex reversals, and Xenopus tropicalis already Valerie tell you about that. And in Xenopus, and the other Xenopus in the lobbies, that we see the testes are immature and they exhibit anti-spermatozoidic cavities. But it's not only these two types of ETCs that produce this kind of problems. We also know, but that several studies with endocrine disruption chemicals, like and all this kind of I show you here types of chemicals can produce different effects relate with the reproduction and gonad development, like the proportion of the sex fractions, gonad differentiation problems, and as well, for example, a lack of secondary sexual characteristic development. But in all these problematics, something that is really important, and maybe already Valerie told a little about this, is there is obviously there is things that we don't know and we don't still understand, and one of these is the mechanistic of how these chemicals can produce all these effects in amphibians in specific. So one of our questions is, in all these kind of problematics, what is or not related the ESRT5 alpha 2, like everybody shows in using one of these when they can prove that this enzyme is involved, but it's no a target that in many of the studies that we do, we take in account. The majority, as Barry said, it was more in the estrogenic pathway. So it's because of that we start to wondering how we can study this, what is the real role of this type of enzyme, and because of that we start to not me, Valerie, decide to start to do the knockout, study this mechanistic through the knockout of this enzyme. For that, and thanks to the MBL in University of Chicago, we can produce the knockout for this enzyme using CRISPR gene editing. And as you see here, so we produce the knockout, and that we have is an insertion of one insertion and 4 deletion, and then in an in silico model, you see here the protein, we have a early stop code and don't produce the total enzyme. So we really think that we have an effect there. So then when we reset the animal, the first thing that we want to check is if we can easily distinguish them between females and males. And with the shape and the size of the animal, it was easily to know if they are females or males. But then when we start to look, one of the secondary characteristics in the males, that is the neutral paths, we compare with the controls. The males and the cows don't have the presence of these neutral paths. When we do histology, like you see here, we see that they have like the development of the gland, but no real pigmentation, but also the number of the glands is low, and it's somewhat that have the pigmentations. So we continue. And then we want to try to do the reproductions, and in this here is when we start to have problem, because after several attempts we don't reproduce this animal. So for that we decide to do the in vitro fertilization, and then we can have the success. We can reproduce the animals with in vitro fertilization, but we note that the percentage of fertilization was really low compared with the control. When we do the in vitro fertilization, we compare the testes between both the control and knockouts, and that we see is the size of the knockouts is bigger. Then we see one of them, we do the histology, and that we note is like as you see here, this is the cyst formation, and here you see the different types, the spermatogenesis, and these little things are the spermatocytes, sorry, I say in Spanish, but these are the spermatocytes, and that we see compared with the knockout, as you see here, is like in the control, they are really packaged and organized, but in the knockout, we don't see many. And also we don't see that they are in this kind of organization. So here we start to see there is something. And then when the time passed, one of the things we start to note is that some of our adult males, 3 years adult males, we see that some one of them start to develop the neutral parts. Then this for us was surprise because many of them in the first year, they don't have anything of, it was like as you see in the photo, nothing. So then with this, because it actually was just one, we say, okay, let's try the reproduction to see what happened. So after we do that, we have to say we do the reproduction using the human curinary hormone, and we have fertilized eggs. Here I don't put it, but we have the eggs. So then with all these eggs, we decide to start to okay, now that we have enough eggs to do the study, we start to looking in the different stages of the animal, in the early development, in the gonal development, the gonal maturation, and the sexual maturity to see what we can find, what kind of phenotype we can find. So first, in the early development, one of the things that because science, the studies of previous studies of Valerie, that we want to see what happened in these stages. So, and is there is a compensation for the other types of these enzymes. So like you see here, in the first stages, we see like the ESRD1 and the type 1 and the type 3 compensating the first part of the development, but not in the last stages, like you see here. And then, for our surprise, it's like the ESRDV that also she sees downregulate when she exposed to the team exposed to finasteride. The ESRDV at the beginning is downregulate, but then is upregulate, and this is kind of a compensated. We don't know. We will think, or perhaps this is kind of compensated the lack of the type 2. And also then we check what happened with the androgen receptor, because like I said before, this is the androgen receptor have a higher affinity for the dihydrotestosterone. So here that we see is again, this is the upregulation, like I guess is trying to compensate the lack of the ESRD2. And then in the more late stage that we evaluate, we see that a down regulation, probably because it's saturated the system, we don't know. But this is super interesting for us because it's kind of like a confirm the previous data that already Valerie explained to you, that there is a role of this enzyme during early development, and it will be continue interesting to looking what is happening there. Then we start to looking in the gonad development. So in the stage of metamorphic climate, when they are already passed through all the metamorphosis, we check what happened in the gonads, in especially here in the testes. As we see, the morphology is compared with the control, we see the testes start to be more tiny. And then we see, we do another kind of cut, the transversal cut, and we see that we have a less formation of the cysts through the gonad and compared with the control. Then we go 1 month post metamorphosis when they start the development. We see in the control the clear formation of the cysts and the start of the spermatogenesis. But in the knockout testes, as you see here, they are formation of the cysts, but they are not happen the spermatogenesis. So continue with this at 3 months post metamorphosis. In some way, some of the individuals, 3 of 7, of the knockouts start to happen. You see, we have compared with the control, it's still tiny, but we see that the spermatogenesis happen. And but compared with the control, it still could be, we need to count for sure, but could be less spermatocytes in the content. Then more surprises, other of the individuals, 2 of 7 of the individuals can, as you see this, can have the shape of the testes compared with the control. But then we check, we found that the presence of oocytes in these individuals. An even more aberrant phenotype we see is this one that is kind of have the shape of an ovary, but also have a lot of oocytes, and that we think is apoptosis also happen. So of these, there are another 2 individuals that have this kind of phenotype. So and again, I showed the previous studies of Valerie and the team. This is like this of a phenotype that we found confirmed that in the previous studies that they see that the finasteride, in special, that is the inhibitor of the 5-alpha reductase, produce the intercept. Here we are confirming with the knockout that this is happened, but also we wonder now, because like I showed you, there is another disease that also produce this kind of phenotypes, and something that we don't know because in these studies was no address is if the ESRD 5-alpha reductase have a role or not. If there is this kind of chemicals are targeting this enzyme or no. So this is something that is for us is so interesting. And then this is the more preliminary data that I have. Is that now that the animals are adults, we try to again do use for the morphological external morphological characteristic separate by females and males, and we see in the controls is very easy to find the females and the males with the presence of the neutral paths, but in the knockouts we can have the females. It's easy to find, but we found males with neutral paths. We look under the microscope to confirm the presence or not the neutral paths, and the males, and they are males that are with the body size we can say yes they are male, but we don't find the neutral paths. And then there is another part of the population of the knockout that we don't determine because it's like probably they are more smaller, they are delaying the development, but we don't see a clear female or males characteristic. So with this, we want to continue study. I will let you know in a moment. But for now, I want to do a summary that until now we know with this knockout model is that during the early development, there is probably a compensatory effect by the ESRD 5 beta reductase. And also during gonadal development and gonadal maturation, we have altered differentiation, sex differentiation. We have a delay in the development. We have a testicular presence in the oocytes, testicular presence. And also we have an intercept phenotype. And in the adult, we see like the bigger testes, a low percentage of the fertilization and a lack of the neutral path development. And as a perspective that we want to address in the future is because here in between this early development and the gonadal development, we have the sex determination period. So we want to check what happened with the compensatory effect in this part. Also, we want to check in all the part that I show you, the histology, what is happening with this compensatory effect, because if you remember, there are some animals that can't develop the testes, so we want to know what is happening with these animals, how they can do, have a nice testes, or why the others that have an ovary don't have a tested. So we want to check also the estrogen receptor signaling, the thyroid hormone pathway, and all these kind of mechanistic things that is happening here to have these phenotypes. And also in the adults, we would like to do the reproduction trying to do different combinations, and especially we want to know, because the low percentage of fertilization, we want to know if this is because it's the male or is because the female that also can have effects in the females. Until now, in all the histology and all this part, we don't see any effect in the ovary. So we really like to address this part as well. So, and that's it. So thank you so much, and thank you for all the team, and in special thanks to she's a PhD now student, and she was a stagiaire that helped me along with all this work, and obviously thanks to Valerie for the opportunity, and that's it.
Thank you very much, Diana, for an excellent presentation. And congratulations to you for the award. So the floor is open for questions again. Maybe I'll get started while we wait for more questions. So in terms of the compensation, so I'm not an expert at all in 5-alpha reductases, and it's complex and in many forms. I guess, are there other activity assays, and do we know anything about, whether the activity of these different forms is similar, even if it's not in frogs?
Yeah, that we know is more for studies in prostate cancer,
Okay,
but it's kind of this happened like they start to when they like Barry say they take finasteride, so then the ESRD2 start to down, and the ESRD1 start to compensate.
okay,
But then it's and this produce in cancer, this produce a more proliferation in the
okay.
tumor. So this is that we know for this kind of compensatory effect. We don't know anything about, because it's really weird that we found that ESRDV start to compensate. This expression is something that is really surprised. We expect more for the ESRD1, but probably is something also for the time. Maybe when I start to do in the part of the development of the gonad, maybe it's the ESRD1. So it's really surprised how can they choose which or what is the enzyme that replace the fusion of the that you
Excellent.
you do
And I
them.
think you described the model and the knockout very nicely, and so very interesting but complex phenotype, of course, as well. I guess one other question I had was, did you think, so I think it's convincing that you have a knockout and it's functional, obviously, on many levels. Have you thought of measuring metabolites, sort of the end product of the 5-alpha reductase, to show if there's compensation going on and if levels change?
Yes, for sure. That is something that we need to do. We I think we will do ELISA kits and all this, but as you know, it's more easy way go first for the histology because it's the more cheaper. But
No,
then
you have
I
to,
guess for sure we do that part.
yeah, but you have to start somewhere, and I think it's very
Yeah,
nice.
like I think when we have all, we pick one stage or with time, and we measure the DHT, and we say,
Right.
okay, look, it's down.
Yeah, okay, fair. Great, thank you very much. Juan has a question.
Thank you. Diana, Agustin, send you many hellos. Just a question. Thank you for sharing your beautiful results. Diana, you know that this alpha reductase is involved in the beginning of the spermatogenesis, especially in the differentiation of the spermatogonian stem cells, or perhaps it's later in the spermatogenesis, or when you see this effect, more, I think that in the effect of the EDC2.
Well, because yes, this is something that I try to looking for another, because until now with the finasteride, with the inhibitor, that we know is they don't produce the infertility, or they don't produce like this kind of a phenotype that we see less spermatogenesis. Like this is something that we don't have the data in that moment. But from the studies of with the genetical disorder in humans, that we know is the thing is in human, the testosterone, in at some point, these individuals have a peak of testosterone and they can reproduce well. But and in the first, before of this peak, it's impossible they can reproduce. So probably it's something between because as you see, at some point, the development of the spermatogenesis happen. So it's really don't know where in which point is the ESRD is doing or no a role in the spermatogenesis.
Are you know where is this gene is expressed in kind of cells?
The gene of the HRD,
Yes.
I think, is in the Leydig, I say well, cells.
Yeah.
Yeah, I think it's in that. Not sure yet.
Okay, great.
Thanks.
Thank you very much. We're right on time, so unless there's any more burning questions, I do have a couple of things to quickly talk about. But thank you very much, Diana. Congratulations again to all the awardees and speakers for an excellent session. Much